In this video lecture, we're going to finish off the second part to Chapter 12, and we're going to look at the adaptive defense mechanisms, and this is a specific branch of our immune system, being that third line of defense once we have that bacteria or pathogen or virus has gone through our first two lines of defenses. Now for this, we're looking at a more specific defense mechanism because we're looking at specific surface markers, which we called antigens. And we have lymphocytes, both T cells and B cells. We have macrophages and antigen presenting cells that assist in this process of recognizing that antigen. Now when we look at this adaptive body defense system, we're looking at specifically we're going to look at antigens, and when we look at an antigen, the very first time it enters into our body is going to get primed, meaning that the second time we see this exact same type of antigen in our body, we can have an amount of stronger reaction to that. And that's really what the whole purpose of this adaptive body defense is for. And there's three different things, concepts and aspects about this adaptive defense system. The first one is that it's antigen specific, meaning that we're only going to act against a particular foreign substance, and we're only looking at one specific antigen, meaning that let's say that we have a bacteria cell that has entered into our body. It can have a flagella, and it can also have glycoproteins, it can have glycolipids, different components and cellular proteins on its cell surface. Now, if it's being antigen specific, that only means that we're only going to respond to one specific type of antigen, one specific type of surface marker. So this antibody that we're going to produce by our own bodies can recognize, for example, this receptor. Another antibody can recognize maybe the end of this flagella. Another antibody can recognize this protein surface marker over here. And so it's only antigen specific, meaning it's only going to recognize a very specific and unique marker. Another thing about this third line of defense is that it's systemic, meaning that if this bacterial infection has entered into our foot the very first time, the second time it can enter into our body, let's say that we ate something that's contaminated with this bacteria. Now it's going to our digestive tract. Well, our lymphatic tissues in our Peyer's patches can also recognize, those antibodies can also recognize these antigens in that area. So it's systemic in that it's not restricted to that initial infection site. So we can also mount a response if it's in our digestive system. Another thing is that it has memory, meaning that the second time we come into contact with this exact same type of pathogen, we're going to have a stronger response to get rid of that bacteria the second time around. If we look at this adaptive defense system, we also notice, because we're talking about antigens, there are two different types of antigens. We have non-self antigens and we have self antigens. And it's really up to our cells, our immune cells, to differentiate and distinguish between something that's considered foreign and something that's considered part of our body. Non-self antigens are any substance that's capable of exciting the immune response and provoking an immune response. So these are molecules and complexes that are not normally present in our body. And this is very important in trying to reduce our immune system, especially when someone's going to get a transplant, whether it's an organ transplant or a tissue transplant or anything like that. You want to be able to suppress your own body's immune system so you can accept a foreign tissue type and not reject it. And now we also have self-antigens, and these are on your own individual cells. And this is really what makes it able for you to accept and reject tissues within your body. And so this is really what restricts all those donors for transplantation. Now, here's your cell, and of course, we'll just say this is a red blood cell within your body. And a great example of this is in our Chapter 10 when we looked at blood. Let's say you're a type AB positive. What does that mean? You have all those surface markers for the A antigen, that B antigen, and the RH antigen on top of your red blood cell. Now, if this is a donated blood, that's not yours, so this is a donor blood. If this person is B negative, of course you can accept it because if you're type AB positive, you can accept anything. But let's say that you want to go in this direction. Can someone with a B positive accept your blood type if you have AB positive? They can't. Why? And that's because you have your A surface antigen and you have this positive surface antigen. So this person's body and your body is going to react very differently because of these surface antigens. And so as long as your immune system is healthy and well, your immune cells will not attack your own proteins, but it can attack, or it can mount and trigger an immune response in another person's body if it's considered foreign, like we just saw in this example. Okay, so here is that one more time, the blood types and how it can be reacted and not reacted as a quick review from that illustration above. Now, another reason why this is important to talk about blood and how this immune response is integrated within this is by looking at these antibodies. Antibodies are really those specific proteins that we generate as part of this adaptive response that are specific to targeting these surface antigens. And why is this important? Because, as a reminder, it's going to bind to these foreign antigens. It's going to cause agglutination or clumping, and eventually we can get rid of this by our body. Now, another reason why this is important is because antibodies, there's a review from last chapter. Why are they important? Because here is a surface antigen that's considered foreign. We can have our own antibody cells bind to these foreign surface markers, and we can mount something like a complement reaction to get rid of it by our body. And so what are the roles of the two cells that assist in this process of getting rid of anything foreign? So we have two specific types of lymphocytes. We have B cells, and these are the cells responsible for all those antibodies we keep on talking about. And we have T cells, and this is a cell-mediated branch of our adaptive defense. So we have cell-mediated, and we have humoral, which we also call this mediated. And that's because it deals with our humors, or that liquid portion, that are filled with all of these antibodies. Now, another type of cell that we have within this early defense, we have macrophages. And these macrophages are necessary because what are they going to do? They're going to engulf anything that's considered foreign or self. So if you have a dead or dying worn out cell, it can also engulf that, as we saw in that last illustration, those Pac-Man cells. It can engulf that cell, and as it chews it up, it's going to present a part of that on its cell surface to these lymphocytes, and then by doing that, it can help in this immune system response. So it doesn't respond to the specific antigen, so it's not going to discriminate between self or non-self, but it will help those lymphocytes that do. Where exactly do these cells come from? This is also a review from chapter 10, and we have what's called the hemocytoblast stem cells, and this is found where? In the adult tissues. Good, it's found within that red marrow, and so it's found at that epiphysis of long bones and also some flat bone regions. So all that red marrow here. From this, we give rise to two stem cell lines. We have lymphoid stem cells and myeloid stem cells. And from this lymphoid stem cells, we're going to give rise to both those B and T lymphocytes. Myeloid stem cells is going to give rise to these monocytes, which turn into macrophages, which serve as our antigen-presenting cell. Let's see how we develop those T cells and those B cells. We're going to start off with this diaphysis, looking at that red bone marrow, and because we're looking at the diaphysis, this is going to be in an adolescent. Now we're going to start off with those hemocytoblasts that form those lymphoid stem cells over here, giving rise to those immature lymphocytes. So these are all naive. They haven't received any instructions yet to respond to any specific antigen. From here, if a cell is destined to become a T cell, it's going to migrate to the thymus, and if that cell, that lymphoid stem cell, is going to become destined to become a B cell, it's going to migrate to the bone marrow. Now here, it's a great way to remember it, T, thymus for T cells, B, bone marrow for those B cells. Both in this thymus and the bone marrow is where the cell gains immunocompetence, meaning that the cell is going to become capable of responding to a specific antigen. let's take a look at what I mean by that. Here is that bacteria cell. We have all of these surface antigens on that bacteria cell. And here, let's say that we have this T cell. We have another T cell that stems from this, becomes immature, and then we develop here within the thymus. Now this T cell is going to eventually form within this thymus receptors specific to this bacteria. And because each of these cells can only respond to one specific antigen, this T cell is going to recognize this surface antigen. This T cell is going to recognize this surface antigen. And so when we say that it's going to become capable of responding to a specific antigen, it's going to be capable of responding to one specific antigen. Okay, so this is what it means by gaining that immunocompetence. And it doesn't receive those instructions until it starts maturing within the thymus or the bone marrow. Same thing here with those B cells. Those B cells also will start forming receptors on its surface. That's going to be specific to the whatever antigen that it can eventually come into contact with. Now you might be asking and wondering, well, in an adolescent, this person may never have come into contact with something like this bacteria before. So how does your body know to actually create these receptors specific to these antigens? And it actually has to deal with your genetics. And so immunocompetence and the development of your immunocompetence all deal with your genetics. So it's all predetermined by your DNA. And why is that? Well, it's all because of your ancestors, and it's all because of where you grew up. And in all those traits, typically these receptors are passed on and they become more expressed if it's something that your parents or your grandparents have become into contact with. And as a great example, you living in Hawaii, you create more of these lymphocytes and receptors that are specific to bacteria found within that region compared to bacteria that are found in Antarctica or Africa, and just because of where you have been. So that's how we can develop the specificity of these receptors on your B cells and your T cells. From here, our cells are still immunocompetent, and they're going to migrate towards all of those lymphoid tissues, specifically being your lymph nodes, and also those MALT tissues. Remember those mucus-associated tissues lining your oral cavity and digestive tracts. Only here once, and this is why it's so important also for you to work out and move your arms and move your legs, is to remove all that extra edema. Push all that edema through your lymphatic vessels, your lymphatic capillaries, and then your vessels, and then screen all that fluid, all that lymph into your lymph nodes, because here is where only when you come into contact, when your lymphoids, your T cells or your B cells come into contact with that bacteria, will your cell gain immunocompetence. That's when it'll only become mature and fully immunocompetent. And that's only when, so as a reminder, only when the B cell or T cell comes into contact with a specific antigen. Does it become mature and become fully immunocompetent? Okay, and so by chance, if this bacteria if you had infection by this bacteria, enters into your lymphatic vessels, you finally screen it here within your lymph nodes, then these cells can come into contact with these antigens, and then by doing that, you're going to end up producing more of this specific type of T cells. If that antigen does not come into contact with this if this T cell or B cell does not come into contact with that antigen, you're not going to produce a lot more of that T cell. So it's pretty much just there, hanging out, waiting, and chilling out until it comes into contact with that specific antigen. Okay, overview again once of all those lymphatic vessels, draining, and the importance of moving your arms, moving your legs, being able to screen all of that lymph into all of these lymph nodes. And once again, it's very important to screen everything, build up that immune response, because as you notice here, once you build up those immune response, and you remember we saw those tonsillitis, inflammation of those tonsils, your lymph nodes get bigger, because what's going to happen, you're going to start producing more antibodies, you're going to produce more T cells specific towards that antigen. And once you do that, it can also circulate, enter back into your right subclavian vein or your left subclavian vein, go back towards your heart, and you can circulate all of those cells within your body. Okay, so and mount that systemic response, and that's how you get that systemic response. You produce all those antibodies, more of those T cells, bring it into your circulation, and run it through your entire body. Okay, and here's that review one more time. Once a lymphocyte is immunocompetent, it will be able to react to one and only one distinct antigen. And all of the antigen receptors on the surface of that cell are going to be exactly the same. And so here's a cell. We're going to have receptors. They're going to look exactly the same. And this was a spherical structure or antigen on that bacteria that we saw in the last slide. And also as a reminder, the lymphocyte can only recognize a part of a foreign agent. And so that dealt one more time, dealing with this surface protein marker and not something like the flagella or this other antigen. Okay, so once again, also very important, and this is all written because, once again, I just want to highlight these key and very important concepts, that it's our genes and not the antigens that determine what specific foreign substance our immune system will be able to recognize and resist. And all dealing with what our ancestors and proteins that are mostly expressed and needed to be expressed by our parents and generations before us. Okay, let's take a look at this role of the antigen presenting cell, and that's because this antigen presenting cell is going to come into close contact with our T cells. And so antigen presenting cells, what are they going to do? Anytime there's something foreign, they're going to engulf it through endocytosis or phagocytosis. So phagocytosis being the endocytosis of this foreign body and then forming this phagosome. We're going to fuse that with our lysosome, and we're going to form this, start degrading both this bacteria with all those lysozymes. We're going to break that down into fragments. The majority of that we're going to release back into our body. It's not going to mount an immune response. But of course, it's also going to present some of that portion of that degraded pathogen on the surface of the cell, and which is why it's called an antigen presenting cell. It's going to present a portion of that antigen on the surface. Now, each of these antigens are not going to be exactly the same. This could be part of that flagella. This can be another surface protein marker. And this could be another surface protein marker. And so this is really key for that immune system because it's presenting different antigens to mount a greater response by the different T cells. So antigen presenting cells are going to engulf those antigens through phagocytosis and present them on the surface where they can be recognized by our T cells. Where do we find these macrophages? We find them distributed through our entire tissue. So looking at our lymphoid organs, our connective tissues. We also find them within our skin. So you remember those dendritic cells that we have we learned in chapter 4? Found within our epidermis. We can reach into, grab anything trying to enter into our skin, phagocytize that, and then bring that into our lymphatic capillary bed and bring it to that closest lymph node for presentation to our T cells. Okay. So key note so far, before we continue on looking at how we form B cells and T cells, is that the immune system responds to threats because the cells can recognize foreign substances in the form of antigens, bind to them, and we can also communicate with other cells to have a much broader specific response in our body. Looking at these two different types of immunity we have, dealing with our B cells or our T cells. We have humoral immunity. This is antibody-mediated immunity. And this is because, as we looked at earlier, it has to deal with antibodies found in the humors or the liquid portions of our body. And we also have cellular immunity, which deals with our T cells, And because this is cell mediated, T cells have to come into contact with those antigens. We can target virus-infected cells, cancer cells, cells that are also donated to us, so anything that's considered foreign. So let's start off by looking at this humoral immunity, and this deals with those B cells. And those B lymphocytes will always bind to specific antigens. So let's take a look here. Okay, so here we have that maturation of all of those B cells trying to gain those immunocompetence. So they're not completely immunocompetent or mature yet because they haven't come into contact with these certain antigens. Now these B cells, these are waiting. We'll just say this is in that lymph node. And as we see here, we've developed receptors on the surface of each of these antigens that are very unique and very specific to certain antigens. So they're all going to be present and apparent within our lymph node. Now here we have lymph coming in to our lymph node, and we have these different antigens that also arrive with our lymph. So here we're going to have our B cells are going to start screening all of these lymph and look at these antigens. Here we have a B cell that has a receptor specific to these antigens. This B cell and this B cell do not have receptors that are specific to this antigen. And remember, each of these receptors are exactly the same. So all the greens are being exactly the same, all the dark blues are exactly the same, and all the light blues are exactly the same. When and only when we come into contact with that antigen will these B cells undergo what's called clonal selection, meaning that clonal selection selects for that specific type of B cell clone. So clonal selection of a B cell is the selection of the specific type of B cell clone that has receptors specific to an antigen that's present present. So here we have activated B cells. We're going to produce the exact same type of B cell that has those surface markers, or these surface receptors, I'm sorry. And now what are we going to form? We're going to form two different types of cells from here. We're going to form plasma cells, and these plasma cells are going to produce antibodies that have the exact same type of receptors that recognize this antigen. And we're going to create what's called memory B cells. And memory B cells are going to create the exact same type of cells that have receptors to the specific antigen. Now, because this is the first time, so this is a first exposure to the antigen. Okay, so this is the very first exposure to the antigen. We don't have a lot of these antibodies within our body, and we don't have any memory B cells at all. So why is this important for our immune response? Well, because the second time, and how does this make our immune response much broader the second time, and we can get rid of our diseases much faster the second time, is because the second time you come into contact with this exact same type of antigen, here's this antigen one more time, you've created so many more of these memory B cells that imagine your response. So before you only had a few of these memory B cells, now let's say you have 20 times as much. All these 20 other memory B cells that now you've created all have these exact same type of receptors. They all come into contact with this antigen, and each of these are going to produce their own plasma cells, which produce their own antibodies. So imagine how much faster you can get rid of something, because you've created 20 times or even more than 20 times the amount of antibodies within your system. And of course, when this comes into contact the second time, you're going to produce more memory B cells and that are going to circulate within your body. Okay. So secondary humoral response, these memory cells are long-lived, and that second exposure, which we just saw, will cause a rapid response. And that second response is going to be stronger because we've created more memory B cells, and longer because now we can produce more antibodies because of all those plasma cells and memory B cells, that can get rid of that much faster. So here is this example of how you can have a faster response the second time around due to your antibody concentration. And here is an example of if that antigen is injected into the body. The very first time you're exposed to that, you're going to have your production of red antibodies. It's not going to be that much within your plasma. And here's over time. So over time, of course, if you don't have that infection anymore, your body's not going to produce any of those antibodies. But then the second time you come into contact with the exact same type of antigen, look what happens. You've created all those memory B cells. All those memory B cells in turn have created all those plasma cells. And what happens? You've also created more antibodies in turn. All those antibodies are going to then come into contact with all those antigens, heightening that immune response. And of course, longer lasting, you have more antibodies within your system, more memory B cells within your system. Okay, so active immunity, this is going to occur when those B cells encounter those antigens and produce antibodies. And how can we produce these cells in general? How can we produce more plasma cells in general? You can do it in two ways. It can either be naturally acquired, in which case you have to have direct contact with a bacterial or viral or virus or any other pathogen. So by doing this, if it's naturally acquired, what are you doing? You're actively causing your body to come into contact with these antigens. You're actively going to cause your body to create those plasma cells, and those memory cells. Or you can also get them artificially acquired, meaning that you can introduce a dead antigen. So maybe let's say you don't want to get into contact with a bacteria, but you can get into contact with a dead or weakened bacteria or virus to stimulate your body to produce more antibodies and produce all of that memory. And this is specifically important during that flu season. This is why people are always encouraging you to get flu shots, because the very first time you come into contact with that flu, that's considered to be the most prevalent one during that winter season or that flu season, you can receive a dead or weakened virus into your body. And then what are you going to do? You're introducing all those antigens. And by doing that, you're going to trigger and cheat, or not really cheat, but you're going to trick your body into thinking that you have that active live virus within your body. So you're going to produce all those plasma B cells and all of those memory cells. So the second time you come into contact with that virus, so second time, what's going to happen? You've already have all those memory B cells and all those plasma cells, and you can recover from that cold or flu much faster. And this is especially important when people say you're in a very active area or a very populated area, especially lots of children, if you're going to school. And that's because you can stop the spread much faster if you have the vaccine versus having to go through the entire phase of getting sick. So that's the benefit of getting vaccinated. Okay, so you can also get these antibodies in two other ways. You can get it naturally, and the only other way naturally to get it is from mother to fetus. And we'll see eventually that the specific antibody for this is IgG. And you can also get it artificially from another person's immune system. So you can also get it from like rabbits. So you can actually inject rabbits with antigens, extract their serum, and isolate their antibodies and inject those antibodies into your body. If anybody ever got a snake bite, also antibodies, if you get to counteract the reaction of that venom, use those antibodies that will bind to all that venom to prevent it from coagulating your blood. So here is a great question. If you receive antibodies, can immunological memory occur? So let's say immunological memory, remember what needs to happen for immunological memory to occur. And that deals with having this memory B cell be able to grow and divide to create more plasma cells and more memory B cells the second time around. And the answer is no, you cannot. And why is that? Because antibodies are going to directly attack and bind to those antigens. The only time you can have immunological memory is if you have direct contact with that antigen. Now this antibodies, what's going to do, it's going to provide you temporary protection because it's borrowed antibodies. It's not your body creating them themselves. And so it's for that temporary only relief or only temporary relief of anything from that pathogen. Here's that overview of both this naturally acquired form or artificially acquired form of immunity. You can have active, naturally acquired immunity, in which case you come into direct contact with that pathogen. You can have naturally acquired passive immunity, in which case you gain antibodies directly from your mother into your fetus. You can have active, artificially acquired immunity, in which case you introduce a dead or weakened virus or pathogen into the body, tricking your body into thinking that it's actually there and then receiving and producing antibodies to that. Or you can have passive, artificially acquired immunity, in which case you're going to receive an injection of immune serum, so antibodies specific to a toxin. Okay, so make sure that you know these 4 different forms. Okay, so looking more closely at this passive immunity, how else can we use it? So not only can you use it for the relief of toxins and trying to get rid of some poisons within your body, you can also use this for research purposes and also diagnosis of pregnancy. And so remember that pregnancy test, HCG, that's what antibodies are used for. Antibodies are used for detecting the presence of HCG. Now, I'm going to go into a little more depth about the research purposes of this, and we can use what are called monoclonal antibodies. And the way this works is that if you have, let's say, an animal, you can use a mouse. Here's the eye, whiskers, tail. What you can do is you can inject a mouse with a specific type of antigen. And this is especially important for research when researchers are looking for specific markers that can be might be implicated in tumor progression and cancer prognosis, and they want to harvest and produce more of this antibodies that are going to react against a specific protein of interest. And so let's say, and I can give you an example of my research, I use a protein RAD51, and this is a double-stranded DNA repair protein. And so what you can do is you can inject antigens. So here's this protein, you can digest it, break it apart into different regions. You can inject all of this into a mouse, and what's that mouse going to do? It's going to, because it's considered foreign, it's going to start creating antibodies to this antigen. And then what can you do after that? Well, it's going to start creating all of these cells within the spleen and this mouse, so you can extract cells from the spleen of the mouse, and you can fuse that with a cancerous cell. So it's called a cancerous B cell. Now, anything that's considered cancerous is going to be uncontrolled cellular growth, meaning that the cell is going to continue to grow and divide and grow and divide and grow and divide. So you can have these B cells, these cancerous B cells, come into contact with these cells from the spleen. And what you can then produce is a large amount of antibodies specific to this protein of interest. Now, you can also do this not only in mice, but people often do it in rabbits. They also do it in dogs, which is kind of sad. Here in the Bay Area, I know I've seen on Craigslist some dogs that were not, thankfully, they haven't been researched on just yet, but they're up for adoption because some of these research test plants decided not to, or continued, or they seized their studies and were giving up these dogs for adoption. So pretty sad, but all in the name of research. So here is an example of how you can use these antibodies for research purposes. And so you can get a section of tissue. We can say this is a cancer tissue section, and you want to determine whether or not there's a high percentage of that protein that can cause and give rise to cancer or not. And so we're really looking for targets. So you can create this antibody, or you use this antibody that's going to look for the specific protein marker. It's going to bind to it. Once it's bound, and if it's bound to that, you can use a second antibody that's going to look for any of this first antibody that's bound to this antigen. You add some reagents, And anywhere that you see this dark discoloration is where you see the presence of this antigen. So lots of steps just for that one little single thing, right? Now, here's a little bit of my research that I did, and it's pretty cool because this deals with oral cancer cells in the human cavity. And so we looked at squamous cell carcinoma from human biopsies, and this is the left floor of the mouth, the gingiva, right lateral tongue, and the tongue. And as I mentioned, I was looking at for the specific protein called RAD51, which we believed was implicated in oral carcinogenesis. So, meaning that we think and we hypothesize that an increase of RAD51 would be expressed if someone has more or has a later stage of cancer growth. And so why is that important? Well, then you can start developing treatments, drugs that target the specific protein and hopefully use that as a therapy to reduce the progression of this cancer. And so anywhere that you see this dark stain is where we stain for RAD51. And it's pretty cool because if you even look closely, you see here this is a cell. What stage of mitosis is this? You see all those chromosomes that are condensed, they're lined up in the middle. That's metaphase. They'll line up right in the middle. Here's another metaphase, a cell in that metaphase state. And so anything that's darkly stained brown is where we see all of that RAD51. So pretty interesting and neat stuff. Now, oh, and also actually a pretty neat thing too, which actually isn't very neat, but we also did oral cheek biopsies in hamsters, which is kind of sad because you induce tumor growth in the hamster cheek pouch, and then you cause them to kind of blow up. And actually, let me pause right now and show you an illustration really quick. Okay, so I pulled up an illustration from one of the researches I've done in the past, and just kind of show you what else is going on there, current research going on. And so here we have Syrian golden hamsters. Half of them received treatment with what's called vitamin D, and half of them didn't. But what you do to determine, we actually wanted to know if vitamin D was going to decrease the ability for cancer progression. And so what you do is you can also treat the inner tissues with this carcinogenic agent called DMBA. Well, I won't have to go into depth about that, but you repeatedly brush over the cheek with this cancer-inducing agent to create all of these cancers. Now, at the same time, we also injected these hamsters with vitamin D to see if that's actually going to delay the onset of carcinogenesis. And after that, of course, you have to sacrifice these hamsters, excise these tumors, and then look for the presence or absence of those proteins of interest. And it's actually interesting because what we did see was this delay in oral cancer progression of the buccal pouch in hamsters that were treated with vitamin D. So pretty neat stuff. Okay, let's go back to lecture. If we look at the antibodies, we have two major regions. We actually compose of 4 amino acid chains. These are held by disulfide bonds over here. We have the variable end, and this is the end that's going to be specific to that antigen. And we have the constant end, and this is going to define the specific type of class of antibody that we're going to be looking at. Okay, so variable end, antigen specific, found at one end of each of those 4 chains, and then constant region, again being that stem of that antibody and determining the type of antibody that's going to be formed. If you look at the antibodies itself, we have 5 major classes, and the way I remember them is this acronym MAG, M for IgM, IgA, IgD, IgE, and then IgE. And all of these antibodies, they have this basic or similar Y shape, but they all have different roles. And so IgM, this is the largest of the antibody classes used in that complement fixation, which we saw before. It's going to recognize any of this surface marker that's considered foreign. Complement proteins are going to come in and then form that MAC complex. IgA, this is found mostly in our mucus and also our body surfaces. IgD, this is going to activate our B cells. IgG, this is going to be the only one, once again, that crosses our placenta. So from mother to fetus, also needed for complement fixation. And because it crosses the mother to fetus, it's involved in this passive immunity. So IgE, this is involved in allergy. So for you getting the allergic reaction, it's because of these guys over here. So we'll actually get into allergies later in this video. This slide really highlights why it's so important that we need these antibodies and their roles within their body. Start off here with this first example of complement. So this occurs during that innate defense system, and it's actively efficient when antibodies are going to bind to these targets. And so here we have antibodies binding to the targets, and of course we form those MAC complexes. Another thing we have here is neutralization. So neutralization occurs when those antibodies will bind to those specific sites on the bacteria cell wall, or those exotoxins. It can either be toxic chemicals secreted by bacteria or even viruses themselves, anything secreted by those. So we can help bind to that and then mask those effects. We also saw here within our red blood cells, agglutination is when those antibodies will bind to more than one antigen that's exactly the same, and then will form this lattice network forms, and we can have eventually phagocytosis of this entire complex. And then finally, we have precipitation, and this involves cross-linking all of these solutes out of the solution, and eventually these will all settle out. And so these three, neutralization, agglutination, and precipitation, these will enhance phagocytosis and get rid of those antigens by our body. Complement will also enhance phagocytosis, but it'll lead to cell lysis, burst open anything that's considered for those cells that are foreign, also leading to chemotaxis. With chemotaxis, we have migration of those neutrophils and macrophages to this area of infection, and we can increase histamine release that causes dilation, leaky vessels to encourage and increase this response of those neutrophils and macrophages to this area as well. Moving on to the cell-mediated branch of the acquired adaptive immunity. This is when we deal with those T cells, and the T cells have to come into contact with those antigen-presenting cells, and meaning that those macrophages or those dendritic cells have to engulf anything that's considered foreign and then present those antigens on their cell surface. And so these T cells are responsible for then recognizing these antigens as being non-self and self. And remember, these macrophages, they're going to engulf and destroy anything that's considered part of your body or anything that's considered foreign. So let's look at how this works. With T cells, we actually have two different types of classes of T cells. We have the first one called helper T cells, and we have cytotoxic killer or cytotoxic T cells. Okay, so how do these work differently from our B cells? Well, our B cells are going to produce antibodies, and our T cells need to come into direct contact with that antigen that can only be presented by those macrophages. So here we have an antigen presenting cell. It's going to present that antigen on its cell surface to this helper T cell. This helper T cell, just like that B cell, can only recognize specific antigens. So once it recognizes a specific antigen, what's it going to do? It's going to recruit the second type of T cell called cytotoxic T cells that have the exact same type of receptor for this antigen. So it's going to recruit that. Cytotoxic T cell, what do they have? They have a mission to kill. They're like the Terminator. Anything that they're told to kill, they will kill, especially if it looks just like this. And so once they receive that signals, it's going to find any antigen that has this specific surface marker. It's going to bind to it, and it's going to directly kill it. And it's by causing that cell to lyse. Now, another thing the helper T cell does, it also releases cytokines. And what do cytokines do? Cytokines are going to call in and increase the responsiveness of more helper T cells, more cytotoxic T cells, and also more of these macrophages to this site of infection. Now, what else do we call in? We also call in B cells. B cells, what do we need B cells for? For that humoral immunity branch we just covered, we need to produce all of those plasma cells to produce those antibodies and those memory B cells. So you can see why these helper T cells are extremely important in this immune response, because they need to target and call in, and they can communicate with so many other types of immune cells. Now, B cells themselves, they can also respond directly to these antigens. They do not need to have and receive signals from these helper T cells. However, because helper T cells are involved in this process, it can mount a much broader, stronger response. Let's take a look now at those cytotoxic T cells and why are they necessary for killing and destroying. And that's because they release these molecules called perforins. And perforins, once they come into contact with that targeted cell, it's going to automatically release these performance, and think of the term perforance, so it's creating pores. And when it creates these pores, we're going to release what are called granzymes. Granzymes are going to enter into that target cell and start digesting and degrading that cell itself inside. And so when it starts degrading the cell internal contents, we can also eventually cause lysis. So with these pores, water is going to rush in and eventually cause that cell to lyse. So more other types of T cells, we have also regulatory T cells. So these are going to release chemicals to suppress the activity of both your T cells and your B cells. And this will help maintain that homeostasis to prevent uncontrolled activity. Now, also a few of these will become memory cells, memory T cells. So the second time we have a response, it'll also be a much stronger response. Here's this overview of both that humoral and cellular mediated branches of the adaptive immune response. And let's go through everything here. And of course, this is something you should know for your exam. Okay, so first you're going to come into contact with that antigen. If there's 3 different things that can happen. Let's start off with this first pathway all the way towards the left. We can have B cells come into direct contact with these antigens. It's going to give rise to both your plasma cells, which will secrete antibodies that have receptors specific for that antigen. It'll also give rise to memory B cells, so the second time you have an immune response, it'll be much stronger. Second thing that can happen with this antigen is we can engulf it by macrophages, and this becomes an antigen-presenting cell. So it's going to stimulate, by presenting that antigen, it's going to stimulate your helper T cells. Helper T cells being critical to stimulating also your B cells, stimulating cytotoxic T cells, and also producing more memory T cells. Third thing that can happen is these antigens displayed by infected cells, they can be activated. So the cytotoxic T cells, we saw that in the last video lecture, can also come into contact with these direct antigens if it has a surface marker specific to it, and also cause it to destroy. And cytotoxic T cells will also give rise to more active cytotoxic cells and also memory T cells. And once again, make sure that you're comfortable with going through this entire illustration and explaining the different pathways for the immune response. Now let's go over the organ transplants and rejection. And so once again, this is the recognition or the non-recognition of surface antigens. And these are the major types of grafts that you can have. You can have autografts, in which case you're just removing one tissue from one side of your body and putting it to another region. And this is common especially in burns. If you want to take tissue from one area, you can do that and bring it to another. Isografts, this is tissue grafts from an identical person, typically an identical twin. Both of these are good donors. Very similar, of course, yourself. You're not going to reject your own body tissue. And an ideal donor being your identical twin, very similar tissue types. Allograft, this is when tissue is taken from an unrelated person. So it's better if you have a close match. So bone marrow transplants, if someone that you know has lymphoma. Xenografts, this is tissue taken from a different animal. These are typically never successful. Although I had one student who said her grandma had one of her valves within her heart replaced by a valve from a pig. So pretty interesting things. Also, right now, I know if you look online, which I can pull up in a little bit too, you can also increase and grow ears, grow different organs in mice. So I saw there's an ear that's growing in the mice's back. I can pull that up for you in a little bit. Now let's look at allergies. And so allergies, these are abnormal vigorous immune responses. We can have immediate hypersensitivity, and this is triggered by the release of histamine, so from IgE binding to cells called mast cells. And these reactions begin within seconds of contact with the allergen, and the second time it can go through your entire body and have the systemic response. This is called anaphylactic shock, which is very dangerous. We can have delayed hypersensitivity. This is triggered by lymphokines from activated T helper cells. These are also going to stimulate monocytes and macrophages. So these are delayed because it occurs 1 to 3 days after you come into contact with that antigen. This is taking a closer look at how we can have that localized allergic reaction and also having an anaphylactic shock the second time you're exposed to that same allergen. And so here's an example, especially if someone who's allergic to like hay or some sort of pollen granules. The first time you come into contact with this antigen, your plasma B cells are going to create these antibodies against this antigen. And these antibodies, we're specifically talking about IgE antibodies, are going to bind to specialized cells called mast cells. Now, these mast cells contain granules filled with histamine, and histamine can do a lot of things downstream. So the very first time you come into contact with this allergen, you're not going to release any of this histamine. You're just prepping these granules to release the histamine the second time you come into contact with the exact same type of antigen. Now, the second time you come into contact with the antigen, what happens? Now all of this IgE that's bound to these mast cells, it's going to bind to these antigens, and it's going to trigger the release of all this histamine from your mast cells. And what does histamine do downstream? Well, a lot of the signaling characteristics of having an allergic reaction, you're going to have edema, meaning you're going to have leaky capillaries. A lot of that fluid is going to come out towards like surrounding tissues and cells. You're also going to have an increase in mucus production, especially within that respiratory tract. And also you're going to have constriction of the bronchioles. And now why is this going to be important and also dangerous? It's because these bronchioles lead to this terminal sac called the alveoli. And these alveoli are where all that gas exchange occurs between your blood and the external environment. And so if these bronchioles are constricted, what happens? You're going to reduce the ability for air to come into and out of your air sacs, and thereby causing you to sometimes even suffocate, right? So if you have a decrease here, and also a lot of that mucus buildup closes off and restricts all the oxygen flow into your respiratory system. So very important to, if you know that you're allergic to something, you prevent or try to restrain yourself from getting close to those antigens. Then also you can take a lot of those medications, so antihistamine. A lot of the medications for allergic reactions are antihistamine. So what does that mean? So that means that anytime histamine is released by these mast cells, these antihistamines are going to bind to this histamine and inhibit it from and prevent it from actually binding to any of these surface markers. So it can either trigger and target these histamine itself, or also the receptors for histamine on these target cell types. So it can block it and prevent histamine from binding to these receptors, causing these downstream effects. And so that's something you also need to know, these antihistamines and also these characteristic symbols and signs of having an allergic reaction. Now, of course, anaphylactic shock, how can that happen? What happens with that? It's having that system-wide response to that allergen. And so it's not just in one area concerning your lungs, it can be found within your entire body. Okay, another thing we're going to cover now are immunodeficiency. So when does this occur? This occurs if and when you have a deficiency with your immune system, in the sense that your immune system does not distinguish between itself and something that's considered non-self. And so typically the body will produce those antibodies towards the certain targets, and also those T lymphocytes will target specifically non-self proteins or antigens. However, with autoimmune diseases, a lot of times these T lymphocytes are going to start attacking its own tissues. And so a great example for this is AIDS, acquired immunodeficiency syndrome. And this is caused by HIV, human immunodeficiency virus. And this virus, so the way viruses work is that because viruses, they don't have any metabolic machinery at all within itself. Meaning that, remember, within our cell, we have the nucleus, we have the ribosomes to produce all those proteins, we have the Golgi apparatus, smooth ER, lysosomes. Viruses themselves, they don't have that. So what they have only is they can have a double-stranded DNA, they can have a single-stranded DNA, RNA, or they can do reverse transcriptase, in which case it'll cause a synthesis of specific DNA from RNA. Now, how does this work? So with viruses, in order for them to duplicate and replicate, they have to attack healthy cells. And so what's it going to do? It's going to pretty much use it and then abuse it and then kill it. And so it's going to attack, it's going to bind to a cell, it's going to enter into the cell, and it's going to use and trick the cell to create a lot more copies of this itself. And once it's done, that cell is going to die, lyse, and it's going to free up and create more of that specific type of virus. And of course, these cells, these virions, can attack another healthy cell and continue to propagate itself in that way. And remember those interferons, they help to interfere with this process of the viruses, these virons, to continue to replicate themselves. And so here, this specific type of virus, the HIV virus, it associates with the CD4 protein found on the T helper cell's membrane. So because of that, HIV specifically targets the T helper cells. Why is that important? Well, once we damage these T helper cells, eventually we can't produce more T cells. And if we go back to this illustration here, we will see how important, once again, these T helper cells are in this immune response. Remember, it helps to stimulate all of this B cells. It stimulates the C cytotoxic cells. It releases cytokines to increase and encourage more macrophages to the site of infection. It also encourages and stimulates the production of memory T cells, which in turn also stimulate the production of more cytotoxic T cells. And so by having the virus attack specifically this T helper cell, we're really decreasing this strong ability to mount this immune response. Now, why is it then that it's called acquired immunodeficiency syndrome? It's called acquired because it all starts with HIV. HIV is always seen as HIV and AIDS because HIV leads to AIDS, in that eventually you acquire the inability to be able to have an active immune system. So it's an acquired immunodeficiency syndrome. So over time, you lose your deficiency of your immune system. Okay, now why is it that eventually you cannot ward off the weakest organisms? Well, if you have a decrease in the ability to call in all of these other cells, what happens then? Then you have a decreased ability to fight off infection. And a lot of times people with AIDS, they end up dying from something so small like a cut, where the bacteria comes into the body and it ends up growing and dividing and spreading because you don't have this great large immune response to fight off that infection. Other autoimmune diseases to be familiar with, we have multiple sclerosis, and this is when the body starts attacking the white matter of the brain and spinal cord. And if you recall the anatomy of a neuron, you have the cell body, the axon, and then the axon terminal. And what's covering this axon are these myelin sheets. Myelin sheets serving two purposes. We have conductivity of that action potential, and so bringing that action potential from your cell body all the way to the axon terminal. But then also we have this protection of this axon itself. And so with multiple sclerosis, the immune system starts attacking all of this myelin sheets. So what happens? You have a decreased ability for communication between your brain, spinal cord, and external regions of your body. And also over time, what do you have? You have impairment of motor control. So what does that deal with? That deals with your skeletal system. Type 1 diabetes mellitus, this is when the immune system starts destroying the beta cells of the pancreas. Beta cells are responsible for producing insulin, and what happens if you don't have any insulin? You're going to have high systemic levels of glucose. And what happens if you have high levels of glucose? Well, glucose is nice and sticky. It can easily catch things, giving rise to plaque buildup within your vessels. Also, for some people with diabetes, what's also common with them is how it affects their eyes. And so this can damage the vessels leading towards the eyes. Patients also have cataracts and glaucoma. Rheumatoid arthritis, this is an autoimmune disease in which case it attacks the synovial fluid found between joints. And we see this here within the hands. Saw this also in chapter 5. All the synovial fluid here found between the different bones. And then it starts producing proteins that the body begins to identify as foreign. And with an increase of serum to this area, increase in white blood cells and antibodies, we have an increase of pressure buildup within these joint cavities and spaces. And so we push out the bones into differing angles, giving rise to the disfigurement that's characteristic of rheumatoid arthritis. Looking at the developmental aspects of the lymphatic system and body defenses, except for the thymus and spleen, all the other lymphoid organs are poorly developed before birth. And the only type of immunity that we can get from newborn, from mother to fetus or mother to newborn, is by passive immunity. And so we have no functioning lymphocytes at birth. Remember, all of those lymphocytes are going to be immunocompetent They're going to be immature, immunocompetent. However, only when they come into contact with that antigen will they become fully mature active cells, right? All active B cells and T cells. And of course, the only antibody crossing from mother to fetus is IgG. Okay? And that's it for chapter 12.